Hello, everyone. Today I will be speaking on the topic how to choose a MASH model mice. This is a practical tips for preclinical trial design. So let's get started. First of all, on this slide, I'll explain why selecting a MASH model is so important at this particular moment in medical world. Please look at the left side. It summarizes the history of MASH drug discovery to this date. Until now, MASH has been considered a very challenging field. In fact, while many drugs have been developed, they failed to demonstrate efficacy in phase 3 trials, leading to a series of development halts and deferred approval. For example, elafibranor failed to meet its primary endpoint, and OCA ultimately did not receive approval. Against this backdrop, MASH has been viewed as a high-risk field with low success rates. However, this situation has changed significantly in recent years. Resmetirom was the first to receive approval, and semaglutide also succeeded in phase 3 trials, has been granted accelerated approval. As a result, MASH has been repositioned as a treatable disease. Furthermore, previous development efforts focused primarily on surrogate endpoints such as NAFLD activity score and fibrosis. In contrast, we have now entered a stage where clinical outcomes such as progression to cirrhosis and survival rates, those are required. In other words, the goals of development themselves have changed. The cardiometabolic criteria are shown here. Since 2021, NASH has been redefined as MUSCLE-D/MASH, and the disease has been classified as one based on metabolic abnormalities. As shown on the left side, the phase of drug discovery has changed, and as shown on the right side, definition of the disease has also changed. As a result, the importance of model selection has increased significantly. In this slide, we'll introduce the latest trends. First and foremost, drug discovery in the MASH field has advanced rapidly over the past few years. As mentioned earlier, thanks to the success of drugs like resmetirom and semaglutide, this field is evolving into a market where the drugs actually succeeded. Within this context, the mechanism of action are drawing particular interest. For example, the focus is on drugs that act on metabolism, such as resmetirom. It is a THR beta agonist. Semaglutide is a GLP-1 agonist and FGF1 agonist. What this reveals is that MASH is, after all, a metabolic disease. In other words, rather than simply targeting the liver, it has become crucial to improve systemic metabolism. There are also significant developments from a business perspective. M&A activity is also intensifying, with companies developing FGF21 agonists, forming partnerships with major pharmaceutical firms, or being acquired by them. To summarize, drug development is accelerating due to the success of these agents. Metabolic targets are taking center stage. The market is also beginning to see significant movement. These are the current trends. Given this trend, the question of which model to use to replicate which disease state is becoming more important than ever. Okay, we'll go to the next page. In this slide, we will explain the diagnostics flowchart for MUSCLE-D and MASH. First, this flowchart illustrates the process for diagnosing MUSCLE-D in patients with confirmed fatty liver. We check for fat accumulation in the liver, then we rule out alcoholic and other liver diseases and assess whether metabolic abnormalities are present. Cardiometabolic criteria shown on the right are used for this purpose. As shown on the slide, the diagnosis of muscle D includes factors such as BMI, blood glucose, blood pressure, triglycerides, and HDL cholesterol, and at least one of these criteria must be met. There's a key point to note here. Criteria includes so-called cardiovascular indicators, such as blood pressure and lipids. This means the muscle D is not just only a liver disease, but also is classified as a disease based on systemic metabolic abnormalities, that is as part of cardiometabolic disease. Next, I will explain the endpoints required by the FDA. Please look at the left side. This shows the evaluation criteria used in the clinical trials for the two drugs approved for this time, resmetirom and semaglutide. The basis for the current approval is a surrogate endpoint. Specifically, improvements in NAFLD activity score and fibrosis were evaluated. Approval was granted based on the confirmation on these improvements. However, this is strictly an accelerated approval. As shown below, clinical trials are still ongoing, and composite endpoints, including progression to cirrhosis and bleeding encephalopathy, are being evaluated. This is a crucial point. According to FDA guidelines, to obtain a final full approval, efficacy must be demonstrated for these clinical outcomes, namely progression to cirrhosis, occurrence of complications, transplantation, and death. In other words, the current MASH development process involves first obtaining approval based on surrogate endpoints and then demonstrating its ultimate value through clinical outcomes. Understanding this structure is crucial when considering what should be evaluated in preclinical studies. Then this slide, I will explain surrogate endpoints. So what exactly are surrogate endpoints, you may ask? They are indicators used in place of final clinical outcomes, such as survival rate or the progression of cirrhosis. In this case of MASH, because evaluating clinical outcomes takes time, surrogate endpoints are used as a primary step as indicators that can predict future improvement. In the current development of MASH, two indicators are particularly important. The first is improvement in NAFLD activity score, and the second is improvement in fibrosis or fibrosis resolution. While NAFLD activity score assesses improvements in inflammation and hepatocyte damage, fibrosis looks at the more advanced condition, essentially how much the hardened liver can recover. Please look at the right side of the slide. This shows the components of the NAFLD activity score. The NAFLD activity score is a score based on these three elements, which is steatosis, fat accumulation, and inflammation and ballooning. By adding up all these scores, we can quantitatively assess the degree of liver inflammation and damage. On the other hand, fibrosis is evaluated using a separate scoring system. Clinically, it is categorized into stages such as F1 to F4, and whether these stages improve is a key factor. How these two aspects are interpreted directly influences the design of preclinical evaluation protocols. We will then discuss the current MASH clinical trial status. This table summarizes ongoing MASH clinical trials. This data is from the clinicaltrials.gov. Please focus solely on the endpoints here in this red area. In phase 2 trials, changes in liver fat contents and blood markers such as ALT, improvements in NASH, and improvements in fibrosis are the primary surrogate endpoint that is used. On the other hand, phase 3 trials, in addition to improvements in NASH and fibrosis, clinical outcomes such as clinical events, event-free survival are incorporated as evaluation criteria. In other words, as a phase progresses, phase 2 evaluates the surrogate endpoints, and then phase 3 evaluates clinical outcomes, forming this structure. This aligns perfectly with the shift from surrogate endpoint to composite endpoints that I have explained so far. In short, this table demonstrates that evaluation metrics are evolving step by step, even in actual clinical trials. This one shows the history of MASH model. This slide lists representative MASH models in chronological order of their development. First, the MCD model was used in 1996. While it could reproduce fatty liver and inflammation, it had a limitation of not being accompanied by weight loss or metabolic abnormalities, making it a model that diverged from human pathology. And next, in 2009, a company developed the STEM model. A major feature of this model is its ability to evaluate the progression from MASH to liver cancer as a continuous process. Subsequently, model development advanced significantly in the 2010s. In 2013, the CDA-HIFA diet model was introduced, became widely used as a model capable of inducing fibrosis in a short period. Next comes the diamond model, was developed in 2016, followed by Western diet plus CCL4 model in 2018, enabling the reproduction of more advanced fibrosis and disease stages. Furthermore, in 2019, the GAN diet model was introduced, establishing a model capable of reproducing metabolic backgrounds such as obesity, insulin resistance. Looking at this chronologically, it is clear that range of pathological conditions these models can reproduce has expanded from simple fatty liver to fibrosis and then to metabolic conditions, carcinogenesis. In other words, it has become extremely important to select the appropriate model based on the specific objective. In the following section, we will take a detailed look at the specific models available, their respective characteristics, and how they should be selected and utilized. Okay, so this slide provides a summary of the evaluable parameters for 4 representative models. First is the STEM model. With STEM model, it's possible to evaluate both improvements in NAFLD activity score and fibrosis. It can detect the onset of HCC at approximately 20 weeks. It also allows for evaluations closely related to clinical outcomes, such as MELD-related indicators and survival. A key feature of this model is its ability to evaluate disease progression as a continuous process. Next is a gun diet model. This model allows for assessments of NAFLD activity score improvement and the fibrosis. It also enables the evaluation of HCC through long-term exposure, though it is characterized by its time-consuming nature. On the other hand, reports on advanced clinical outcomes such as ascites and encephalopathy are limited. Next is a CDA-HIFA diet model. While this model is suitable for evaluating fibrosis, it is not suitable for assessing NASH improvements because it is difficult to evaluate certain aspects of the NASH, particularly the ballooning. Furthermore, while HCC can be assessed, it requires a long period of time and the evaluation of clinical outcomes is limited. Finally, there is a Western diet plus CCL4 model. This model allows for the assessment of NAFLD activity score and fibrosis, particularly progresses relatively quickly. HCC can also be assessed in a relatively short period. Regarding clinical outcomes, however, while some conditions, such as ascites and portal hypertension are reproduced, overall evaluation remains limited. As such, the parameters that can be evaluated and their scope differ from each model. The STEM model is characterized by its ability to provide a comprehensive end-to-end evaluation covering everything from MASH to all the way HCC and even survival. On the other hand, the other models excel in evaluating specific pathologies or parameters. Therefore, it is important to select the appropriate model based on the specific evaluation objectives. And from here on out, we'll introduce each model, and first we will explain STEM model. This model reproduces the progression from fatty liver to MASH, fibrosis, and ultimately liver cancer, hepatocellular carcinoma, as a continuous process by combining STZ administration with high-fat diet in newborn mice. One of their key features is a rapid progression. It is characterized by the fact that it progressed to HCC within a relatively short period of time, approximately 16 to 20 weeks. Pathologically, findings characteristics of human MASH are also reproduced, specifically ballooning degeneration of hepatocyte. Perisinusoidal fibrosis is also observed. And at the molecular level, changes in markers such as CK18, glypican-3, AFP have been confirmed, making this model that correlates well with human pathology. Another major feature of this model is its ability to evaluate indicators closely related to clinical outcomes, such as ACC and survival rate. As explained earlier, in current drug discovery, clinical outcomes are ultimately prioritized. In that sense, STEM model is extremely useful for the purpose of capturing the changes closely related to clinical outcomes. This model uses pregnant mice sourced from the Jackson laboratory. Next, I will explain the results of evaluating STEM model against cardiometabolic criteria. This slide shows the extent to which the STEM model aligns with MASLD diagnostics criteria explained earlier. As shown on the slide, the STEM model meets the criteria for the following parameters: fasting blood glucose, hemoglobin A1C, serum triglycerides, blood pressure. In other words, we can see that STEM model reproduces multiple metabolic abnormalities, including glucose metabolism, lipid metabolism, blood pressure, within the cardiometabolic criteria. On the other hand, features such as obesity, hyperinsulinoma, anemia are not strongly present, indicating a condition closer to a so-called lean type MASH. Even within the same MASH category, patients with severe obesity and insulin resistance may have different clinical presentations and drug responses compared to lean patients with a history of diabetes. In this context, STEM model can be positioned as a model that replicates lean type MASH centered on glucose metabolism abnormalities. In this way, by organizing the data according to cardiometabolic criteria, we can clearly identify which patient profile the model corresponds to. Next is a STEM model case study. This slide shows the example of a study which the STEM model was actually used for evaluation. In this study, the subjects were divided into 3 groups: the vehicle group, resmetirom group, and the semaglutide group. Administration began at 9 weeks of age when fibrosis was progressing. Evaluations were conducted over a 3-week period through week 12. Evaluation criteria, NAFLD activity score, and fibrosis area. As a result, improvements in NAFLD activity score and fibrosis were observed for all drugs compared to the vehicle group. The STEM model allows for the simultaneous evaluation of NAFLD activity score and fibrosis. This can be linked to future evaluations of HCC and survival. We have compiled a collection of STEM model results summarizing this trial data. It includes trends by target and modality, as well as specific examples of study designs and evaluation criteria. We're currently distributing it to those who request it, so if you're interested, please feel free to contact us. Next, we will discuss CDI-HIFA diet model. This model is designed to induce steatohepatitis with fibrosis by subjecting mice to conditions of choline deficiency, HIFA diet, and low methionine diet. Key features of this model is a rapid progression of fibrosis. In fact, distinct fibrosis develops within approximately 12 weeks, making it highly suitable for screening antifibrotic drugs and demonstrating proof of concept study. Furthermore, because the study period is relatively short and reproducibility is high, it is a model that is easily handled in practical applications. As shown in the slide, the progression of fibrosis can be clearly confirmed using indicators such as increase in the area of fibrosis, elevated serum ALT, and changes in liver weight. Thus, the ability to reliably evaluate severe fibrosis over the relatively short period is a key feature of this model. This model has a clear limitation, but we will discuss that here. Specifically, it exhibits behavior that differs from the metabolic phenotype of human MASH, such as weight loss and reduced blood lipid level. From this perspective of cardiometabolic criteria, this model cannot be said to fully replicate the patient background. Therefore, it's important to use a CDI-HIFA diet model with a clearly defined purpose, specifically as a model for evaluating fibrosis. For the evaluation of metabolic drugs or translational validation, this model alone is insufficient. Its use must be combined with other models. By limiting its application in this way, it becomes a highly useful model. And this is a case study for CDI-HIFA diet model. It shows the example of efficacy evaluation using resmetirom. The study design involved administering resmetirom for a set period while the subjects were fed the CDI-HIFA diet with evaluations conducted at 6 and 12 weeks. Evaluation parameters, liver to body weight ratio, alpha-SMA positive areas, fibrosis area, all of these are indicators used to assess the progression and improvement of fibrosis. As a result, improvements in these indicators were observed in the resmetirom treated group. It confirms its anti-fibrotic effect. In particular, the reduction in alpha-SMA positive area and fibrosis area clearly demonstrates its anti-fibrotic effects. CDI-HIFA diet model is a system capable of evaluating fibrotic improvement effects of drugs such as resmetirom within a relatively short period. On the other hand, as I explained earlier, there are limitations to the reproducibility of the metabolic background in this model. When conducting translational evaluation, it is important to combine these models with others. Next, we'll go through a GAN diet model. This model induces the hepatitis through high-fat, high-cholesterol diet. As shown on the slide, key components of NAFLD activity score, fat accumulation, inflammation, ballooning, and fibrosis are reproduced in a well-balanced manner. For this reason, it is a model well suited for evaluating improvement in MASH. However, there are some points to note. The progression of fibrosis and HCC takes a relatively long time. It requires a long-term exposure. Consequently, rather than being suited for short-term evaluation, this model is designed to assess drug efficacy while monitoring the progression of the disease. The GAN diet model is well suited for tracking the progression of the disease over time while comprehensively evaluating pathological changes. The GAN diet model from the perspective of cardiometabolic criteria. Here, it shows the metabolic profile that the GAN diet model reproduces. The GAN diet model demonstrates the weight gain, impaired glucose tolerance, which is shown as an abnormal OGTT, and elevated blood triglycerides. These correspond to elements included in the cardiometabolic criteria, indicating that the model simultaneously reproduces multiple metabolic abnormality. Therefore, this model exhibits characteristics similar to the pathology of MASH, which is associated with obesity and insulin resistance. While the STEM model represents a lean phenotype, GAN diet model can be positioned as a model that reproduces a condition accompanied by more general metabolic abnormality. In this way, by organizing the data according to cardiometabolic criteria, we can clearly understand the differences in patient backgrounds across the various models. Next, I will explain the wild Western diet plus CCL4 model. This model induces liver damage and promotes fibrosis by administering CCL4 in addition to a Western diet. Elevated plasma ALT and AST level, changes in NAFLD activity score, increased fibrosis area are all indicators that can be used to evaluate the disease state. Key characteristics of this model is the fibrosis progresses relatively rapidly. Consequently, it allows for the efficient evaluation of moderate to severe fibrosis. The Western diet plus CCL4 model is designed to evaluate disease states centered on fibrosis within a relatively short time frame. As such, it is well suited for evaluating advanced fibrosis and for testing antifibrotic drugs. Western diet CCL4 model from the perspective of cardiometabolic criteria. This slide illustrates the pathological backgrounds of the Western diet CCL4 model. Fasting blood glucose and other specific parameters are observed. However, sufficient reproducibility has not been reported for multiple cardiometabolic indicators such as blood pressure and dyslipidemia. This model is designed to promote fibrosis based on exogenous liver injury induced by CCL4. Therefore, the focus of the pathology lies more in liver injury and progression of fibrosis rather than in metabolic abnormalities. It's in this respect, its positioning is different from that of the STEM and GAN diet models. In other words, it is more appropriate to consider the Western diet plus CCL4 model as one that replicates disease stage with advanced fibrosis rather than as a model that replicates patient backgrounds from the cardiometabolic perspective. By organizing information in this way, we can clearly distinguish which patient groups and disease stages each model corresponds to. HE staining allows us to confirm the basic pathological findings of NASH, primarily fatty infiltration, inflammation, and ballooning. Looking at each model individually, in a stem model, small droplet fat infiltration predominates, and ballooning, degeneration, inflammation are clearly evident, revealing findings similar to those seen in human MASH. In GAN diet model, large droplets, steatosis, and inflammation are widely distributed, with each finding appearing in a well-balanced manner. In CDA diet model, steatosis and inflammation are present, but ballooning is absent. Pathological characteristics are different. In the Western diet CCL4 model, small droplet steatosis predominates and ballooning inflammation are clearly observed, confirming findings similar to human NASH. However, these findings are not as pronounced as in the STEM model. This is a stain used to evaluate fibrosis. Differences in the distribution and severity of fibrosis can be observed across the model. In STEM model, fibrosis is observed around the central vein and spread as the disease progresses. F1 to 2 equivalent in clinical trials. In the CDA diet model, relatively severe fibrosis is evident at stage F3. In GAN diet model, it is between F2 to 3, and the Western diet plus CCL4 model at stage F3. Next, we can discuss the approach to model selection. This slide represents a flow chart that outlines how to select a model based on the information covered so far. The first step is to determine which endpoints you wish to evaluate. If you want to evaluate outcomes such as NAFLD activity score, fibrosis, HCC, survival, the STEM model is an appropriate model. Next, if such a comprehensive evaluation is not necessary, the choice depends on which patient profile is being considered. If the patient profile includes obesity and insulin resistance, GAN diet model might be the appropriate choice. This model allows for balanced assessment of the various components of NAFLD activity score, such as fat, inflammation, and ballooning. On the other hand, the STEM model is suitable when assuming a lean phenotype or condition with a diabetic background. Furthermore, if you wish to assess the progression of fibrosis over the short period, Western diet plus CCL4 model could be your option. This model is characterized by its ability to evaluate moderate to severe fibrosis in a relatively short time frame. Additionally, CDA diet model is suitable when the focus is specifically on evaluating fibrosis itself. Thus, by considering these two perspectives, which endpoints to observe and which patient backgrounds to assume, it becomes possible to select the appropriate model. And finally, I would like to explain the importance of model selection. As you have seen so far, each model has its own distinct characteristics and strengths. Key point is that there is no such thing as a one-size-fits-all model. Therefore, it is necessary to select a model by working backward from objective, taking into account the clinical target, disease stage, patient background, and the drug's mechanism of action. Furthermore, understanding the endpoints required by the FDA, namely both surrogate endpoints and clinical outcomes, will clearly clarify the design of preclinical evaluation. Models such as STAM, CDA hypha diet, Western diet, and CCL4, and perhaps the gun diet model each have strength for addressing different research questions. Therefore, rather than relying on a single model, it's important to enhance the connection to clinical practice by combining multiple models as needed. In summary, model selection is not merely a choice of methodology, but a critical element directly linked to the development strategy itself. Appropriate model selection enables more accurate decision making. In this last page, we have our contact information here. At SMC Laboratories, we provide preclinical efficacy testing services, focusing primarily on the fields of inflammation, fibrosis, and cancer. We support the entire process from model selection and study design to histopathological evaluation and data analysis. Regarding the MASH model introduced today, we're excited to hear from you and can consult on matters such as which model to select or which endpoints to evaluate. Additionally, we are happy to provide a portfolio of results for the STAM model, which was mentioned earlier to anyone who is interested. We can also provide proposals tailored to your specific project, so please feel free to contact us if you're interested. Thank you very much for your attention today. We'll see you soon.